Changes in the pigment of photoreceptors stimulates the creation of a nerve signal in the retinal nerve fibers. We will use rods as our example, but although it's not known for sure, cones likely work the same way. In the dark, rods steadily release the neurotransmitter glutamate into a synaptic cleft with a bipolar cell. But when a rod absorbs light, they stop. Rods contain a pigment called rhodopsin. This is also called visual purple. It's a combination of two molecules called opsin and retinol. In the dark, the retinol molecule has a bent shape called cisretinol, but when it absorbs light, it is transformed into a straight form called transretinol. This form then breaks away from the opsin, leaving the opsin colorless instead of violet. For this reason, the process is called bleaching. The opsin triggers a reaction that causes the rod to stop secreting glutamate. Some bipolar cells are inhibited by glutamate, and they're excited when its secretion stops. These cells are therefore excited by rising light intensities. Other bipolar cells, however, are excited by glutamate, and therefore they respond when light intensity drops. When excited, these bipolar cells synapse with the retinal ganglion cells of the optic nerve, and they create a nerve signal that travels toward the central nervous system. As your eye scans a scene, it passes areas of greater and lesser brightness. Their images on the retina cause a rapidly changing pattern of bipolar cell responses as the light intensity on a patch of retina rises and falls. This helps your central nervous system get the data it needs to perceive a proper image.